Most of my interest in the lab is in studying the roles of epigenetics and microbiome in autism. However, I want to speak today about different projects that we have in the lab, and basically talking about looking at using animal models. So now we're going to switch from the people for a minute to the animal models to try to understand some of the molecular mechanisms that underlie some of the more comorbid or non-core behaviors of autism. So we're all familiar with the core phenotypes of autism and how we diagnose by social communication deficits and restrictive behaviors, etc. However, there are many other phenotypes that are often present that oftentimes the families very much want to address, and we need to understand more about the molecular mechanisms underlying those, I'd call it non-core phenotypes. So I'm going to talk about just two projects that we've had in our lab, trying to use mouse models to understand the molecular mechanisms behind these phenotypes. And one of them, we talk about impaired risk assessment or impulsive behaviors. And then the second one about gastrointestinal issues. So in the first study, we used 4 mouse models, 3 of them being genetic. That doesn't, do you see that? Do you see that? No, okay. 3 of them being genetic mouse models for autism, when each mouse model, a gene has been deleted or mutated that is involved in autism. And we wanted to phenotype all of these mice to see if we can find behaviors that are in common in all of these mouse models and have molecular mechanisms that can explain these common phenotypes that are present in all of the mouse models. So at first, we're actually interested in looking at mechanisms behind increased anxiety. And here we ran into an interesting issue. One anxiety test, I don't know if you can see up here for autism, is you put a mouse inside a elevated plus maze. We have closed arms where a mouse can go to feel protected, or into an open area which is rather high so you can look around. And an anxious mouse will want to feel closed and protected, or a non-anxious mouse will go out into the open and explore around. And we found in all the genetic mouse models of autism, all of the models wanted to actually be outside more. In other words, they looked to us less anxious. That didn't make much sense to us compared to what we know more or less in the field. There was another test, we did this called a dark light test, you see in the bottom, where you actually see how much time the mouse wants to be inside a closed area or inside an open, very highly lit area. And we found that 2 of the 3 genetic models wanted to actually be more outside, appearing to be less anxious. We didn't understand how to explain that, and we looked at the videos a little bit more closely. And what we noticed is that in the autism mouse models, what they would tend to do is you'll go to, let's say, the border area. I can't really see that here, but where the border to the open zone, to where they would, which are all open, and they would just jot out there, like just go really fast without looking left or right. So what a mouse usually does when it goes into that area is it'll look and pretend I'm a mouse for a minute, and they'll look left, look right, and then they'll make a decision. Sometimes they go back, sometimes they go forward. When you looked at the videos, we just saw them just going without that risk assessment sort of behavior. So we wanted to quantify that in some method. So we actually used a quantification method, which was made by Maya Lebeau in a previous lab that I worked in, where they actually looked at the risk assessment area in the center of the open arms. In fact, we found that the genetic mouse models spent less time in that risk assessment zone, we call it. Another thing you see on the right side, that is what we call stretch attend. That's when the mouse goes like that, goes back down. It's stretching itself to see its environment. And the genetic autism models are doing that much less. So you seem to have this sort of phenotype, and we quantified this in the dark light test as well. We had a phenotype where the mouse looked like it wasn't assessing any sort of risks, which you can sort of compare that to an impulsive type of behavior. I'll just mention that I also work a little bit on postmortem brain studies. And very often when you get so why did the person pass away, and we're talking about people who are between 6 and 20 years old, when we get the lists from people who are diagnosed with autism, 80 to 85% of them were drowning. And many of those cases that we looked into are cases where they really went close to a river, did not assess the risks of being next to a raging river, and eventually drowned in the end. So this is something that actually has some very much importance in the field. So we were interested in looking what's the molecular mechanism involved in this. So we focused on an area called the dorsal striatum because it's an area involved in decision-making processes. So what we did is we actually took from all of these mouse models, we took out the dorsal striatum, this area of the brain. We did something called gene expression analysis. I'm not going to go too much into all this, the technical stuff. But so I'll just show you basically what was the results of this analysis, which Professor Sergei Schiffman here helped us as well in the analysis. And so we found that all of the genetic mouse models, we can actually correlate between the amount of time that they spent risk assessing to the levels or the expression of genes involved in glutamate signaling in the striatum. So it's just a list of genes involved in glutamate signaling. Glutamate is an excitatory neurotransmitter, which is also found in the striatum in many other areas. And dysregulation in the system, we could correlate directly to the amount of time that the mice spent assessing risks. We further did one other molecular type of analysis and found, and we purified the synapses from the striatum. In fact, found less levels of the receptor of glutamate in the synapses of these mice, in the dorsal striatum. So this gave us some confidence that, in fact, perhaps this is a molecular mechanism which is somewhat involved in this risk assessing behavior. So we used a pharmacological tool to really try to understand this. We gave mice, now this is not autism mice, these are just regular mice. Not autism mice, but not autism model mice. It's just regular mice, give them into the dorsal striatum a pharmacological inhibitor of these same glutamate receptors that we saw were dysregulated before. And we wanted to see what did this in fact cause the same phenotype as we see in the autism model mice. So you inject into the dorsal striatum this, it's called NBQX, a pharmacological inhibitor of specific glutamate receptors. And in fact, what we find is that the mice, after we're giving them this NBQX, they again show this less risk assessment behavior. They jump more out into the open arms. They don't do as much stretch or tend posture. They just sort of become more impulsive-like, as we saw in the autism mouse models. So again, this gives us some feeling that, in fact, this is a mechanism involved in this phenotype. And this is, again, a dark light test, the same thing where we give them this NBQX. They jump out into the open area more, spend more time there, spend less time assessing risks. So overall, from this study, we are concluding that we can have that perhaps the dysfunction of glutamate signaling specific in the dorsal striatum is involved in a dysregulation of risk assessment in individuals with autism. This could be related to certain behaviors of impulsivity that we see. So that was one project. And the second project, we were talking about gastrointestinal issues. Now, for those who are in this lecture this morning from Professor Buchsbaum, so he talked about two sets of genes that are often mutated in autism. One involved in synaptic functions. Another one, genes that are involved in gene regulation. So CHD8 is maybe the top gene of the type that regulates genes. CHD8 is a gene which is highly associated with autism, one of the top genes. And its function is to sit on the DNA and regulate the expression of other genes. And it is found in all of the body, not only the brain. It's found everywhere. It has been shown that in people who have this particular mutation also show clear gastrointestinal symptoms, about 90% of them. Some of them from constipation to real inflammation to all sorts of different symptoms. So we want to understand and take the mouse model of the CHD8 haploinsufficiency and see, can we understand what's going on in the gut specifically, what's the gastrointestinal issues it is causing. So the first thing we did is we took mice that are haploinsufficient for this gene. They have one allele missing. And we give to them into their gut basically a protein which has a fluorophore attached. And the reason we do that is to check how much of it goes into the bloodstream. So there is a condition which is called leaky gut. where you have too much, your gut is basically too leaky and allowing too many things that you ingest to go into the bloodstream. In fact, we find that 2 hours and 6 hours after giving this protein into these mice, when we check blood plasma levels or plasma levels of this protein, they are increased. And in these particular mice. In other words, they have a leaky gut. They're allowing too many things from the gut to go into the body, into the blood system. So our next step was to understand, okay, what's actually the dysfunction in the gut? What's CHD8 doing there? And could this be important for anything really? So it's just a slide showing a bunch of technical stuff, but the idea is that we take the colons, take the intestines from the mice of wild type and knockout. We take the epithelial cells, purify them. Those are the cells which are our defense in the gut, which is inside the gut and in the bloodstream. And we see if there's differences in the gene expression between normal mice and CHD8 haploinsufficient mice. And this slide, actually, for my opinion, is really the most important of anything to talk about, because what we found is that in the gut of these mice, we had nearly 1,000 genes which are differentially expressed in CHD8 haploinsufficiency. Why is that interesting? When we look at the brain of the same mice, about 10 genes change. Very little. In other words, this is one of the top genes for autism, and when we look at what happens in the brain in terms of gene expression, it has very subtle effects. In the gut, it has very large effects. So we just looked a little bit more into what in fact these effects are. We found there's a particular type of cell in the gut called a tough cell, which is involved in our immune response and in gut permeability, which is dysregulated, downregulated a lot in these haploinsufficient mice. And one other interesting thing we found is there was about a 50-fold or 40-fold increase in the amount of antimicrobial peptides in these mice. Or expression at least of these peptides in these mice. What does that mean? These are peptides who their role is basically to kill off particular bacteria that are in our gut. And they're extremely found at higher levels in these haploinsufficient mice, which again will show us some connection between the genetic susceptibility to autism and how it can affect actually even microbiome and levels in the gut. I would just mention, even though I don't show it here, but we are doing studies on microbiome in humans, in individuals with autism. And from some of the preliminary studies we've done, we see that usually there are decreases in good microbiota in individuals with autism. In other words, the problem isn't increases, but actually decreases. And it goes along well with what we see here in terms of increase in antimicrobial peptides. I just mentioned that slide that we see what we're looking at now is in actually the blood of these animals, because if they have a leaky gut, that means something is probably leaking into their bloodstream that could affect their overall physiology, perhaps get into the brain as well. We find that they have a very different metabolitic signature in their blood. In other words, they have a lot of metabolites which could be coming from the gut as well, which is changing between these mouse models. And we're now trying to understand, first of all, what are these metabolites and how they might even affect brain physiology as well. That's it. And of course, I want to thank a lot of wonderful people who work on these projects. And thank you.